Well, the first thing I'd like to do is just to thank the organisers for giving me the chance to present this meet. And what I'm going to do is to move away from some of the lovely material that Chris presented in his talk on in silico production of synthetic DNA, and eventually trying to obtain a plant which can directly utilise diatomic nitrogen, and rather move towards the more practical aspects of how we can manage agricultural outputs in terms of our choice of microsymbionts, and coming back to the genomes and beginning to understand what our choice can be based on, with particular emphasis on some of the adaptive parameters which dictate the biogeography of bacterial species. And the first thing that we need to consider is how important nitrogen is. And we look in an agricultural setting, in quite often impoverished soils for nitrogen, if we provide an input of N, we get a significant increase in crop and pasture production, and that increase in pasture production often leads to increases in animal production. And we've got a real problem coming up in terms of population increases. So we're looking at, in 2050, an increase to about 10 billion people on this planet, and increasing to about 2100 to about 16 billion people. So that's going to place tremendous demands on our resources in terms of food production, and also importantly in terms of delivery of that food. So what I've done here is just on the right-hand side, just shown a density map, and this is just the density of people. I want you to concentrate on the US and that little region on the eastern side, and I'm going to come back to that in a later slide. And we're going to look at the impact of high density populations in one area and the impact that they have on the environment. So we've talked about the fact that nitrogen is absolutely essential for life on this planet. There's two basic sources that we can obtain that nitrogen from. The first is from a non-biological source, such as the chemical synthesis of ammonia through industrial processes, or the Haber-Bosch process, which is the most widely applied process. And from a biological perspective, to produce fixed nitrogen through processes which are in prokaryotes, which are called the diazotropes. And you can see the contributions in terms of a non-biological to a biological context. Here we're looking at an increase of almost twofold when we're looking at the amount of nitrogen which is fixed in a biological context in comparison to a non-biological one. If you put that more into perspective, you can see that when you break this down in a biological system, that most of the amount of N which is being fixed is actually coming from biological nitrogen fixation. And of course, in an agricultural context, it's the symbiotic diazotropes, which are the ones which interact with the legumes and cause that nitrogen to be fixed. And there's a number of ways that these symbiotic bacteria can actually interact with their hosts. And these are just some of the currently known models for that infection process. The most widely known is the interaction of the root nodule bacteria. So after the plant produces exudates, those exudates are recognized by the rhizobia. Those rhizobia can produce nod factors. Those nod factors cause a change in the plant root system, in the root hair. They cause the actual root hair to curl over and entrap those bacteria. And then those bacteria start to invade, forming infection threads, and they can channel down in through the root hair, down into the cortical cells, whereby you get the differentiation of the free-living bacterial forms inside the cortical cells of the plant in vessels which we call symbiosomes. And in those symbiosomes, they differentiate into what we call bacteroids, and there they fix nitrogen. So it's a very intricate dialogue between the plant and the rhizobial partner, and that's the most widely studied process. The other process which they can infect legumes is by a process called crack entry, shown on the left-hand side here. And in crack entry, this is also mediated by nod factors, which are produced in response, once again, to the exudates which are produced by the plant. So these bacteria can enter through the epidermal cracks, and once again, through the invasion process, establish the infection threads, all mediated via the production of those nod factors. Just recently, there was a bit of heresy in the field because there was a report out that, in fact, nod factors do not need to be produced by a very specialised group of the rhizobia, and these are the photosynthetic bradyrhizobia. And it's now been found that in this particular case, these rhizobia infect by the crack entry route, but instead of producing nod factors, they're now producing cytokinins. And these cytokinins come in at the lower part of the signal transduction cascade. So you can see the nod factors coming at the top, causing a change in calcium concentrations or calcium spiking, which initiates that root hair curling and the infection thread development. Here the cytokinins coming much lower than that, but still in the same pathway, leading eventually to increases in gene expression and cell division in the actual plant, and eventually through to the nodule formation. The net result in each of these situations is the formation of the nodule, the intact nodule containing all of those symbiosome with the differentiated bacteria, and it's those differentiated bacteria which can fix the diatomic form of nitrogen through to ammonia and eventually into, by protonation, ammonium ions. Finally, that will be delivered to the plant in the form of an organic source of nitrogen. Okay, so the other important thing I want to stress here is the importance of symbiotic nitrogen fixation in terms of environmental context. And really, symbiotic nitrogen fixation alleviates a lot of the problems that we currently have when we're starting to apply various industrial processes to manufacture the fertilizers that we need to drive productivity in our agricultural systems. Many of you will be familiar with the Haber-Bosch process. Here we combine nitrogen with hydrogen in a direct ratio, and we heat the reactor up to about 940 degrees Fahrenheit or 450 degrees Celsius. And in the process of this heating, we drive off ammonia, but also we generate a lot of greenhouse gases, including a lot of carbon dioxide. The gaseous ammonia can be condensed and liquefied down to liquid ammonia, and that can be fed into nitrogenous fertilizers. When people start to apply that liquid fertilizer, there are all sorts of consequences, and one of the biggest consequences is the release of nitrous oxides and the emissions of those into the environment. And here's just a little caption for those predominant emissions in the US, and you can see by far about 65 to 68% here being contributed by agricultural fertilizers which are being applied. So that's a very big problem, is the manufacture of greenhouse gases. And we talked about population densities on the previous slide, and here you can see the impact. We have very high population densities. You have a lot of production of foods. You have a lot of utilisation of nitrogenous-based fertilisers. You get a lot of deposition of nitrates and ammonium, and eventually these leach out. And so there's a big concern at the moment here for all of those compounds leaching out into the associated waterways and the surrounding environments. So by using SNF, we can circumvent all of these issues. Is it a perfect system? No, absolutely not. We've got to closely match the microsymbionts to the legume hosts, and that's a big problem. Putting this into perspective in a global context, there are large tracts of agricultural soils which are infertile, quite often very high in terms of saline content, also quite often very acidic, in the range from about pH 6 down to about pH 3. And certainly this is the case in Australia, so to bring it back into an Australian setting, this is an example of an arable landscape in the southwestern corner of WA. So this is Western Australia down here, and this is where I'm from, at this little spot down here. You can see the nature of these sandy types of soils, normally red, very salacious, high in salacious content, the water tending to bead on the top. These soils will be very high in saline. You can see the massive amount of clearing that's occurred, and that clearing rising the water table, bringing up the salts with that. And now this is a forecast for increases in salinity, and in the next 20 years, it's been forecasted in the southwestern corner of Australia that we may have 6 million acres of land which is going to be directly affected by high salt concentration. So we've got a real problem there. On top of all of that, we have very high, large areas of very acidic environments. And this all produces a real issue to establishing a very functional, a very efficient symbiotic nitrogen fixation process. On top of that, we've got the issues with climate change. So this is a modelling system which has been set up by the Commonwealth Science and Industry Research Organisation in Australia, and they've just tried to model what will happen to the climate over time, and they are predicting by 2050. So you can see these orange areas here. The more intense the orange colour, the more the change in the temperature. They're predicting that by 2050 that we'll have a 4 to 6 degree temperature rise in the southwestern corner of Australia. So that coupled once again to the existing problems we've got with high temperature and low rainfall means we've got a real problem establishing symbioses in these areas. And we certainly managed to succeed in that area. What we've done is to begin to target germplasm from countries which have a climate which is very similar to ours, but we not only need to consider climate, we also need to consider some of the edaphic variables that go with that. And one of those most important edaphic variables is soil pH, and in particular acidity. So we sourced isolates and our legume germplasm from all around the globe, particularly from the Mediterranean basin and lately from Africa. They've been very valuable in terms of establishing large tracts of very productive land containing very effective symbioses. And by using this approach, we've been able to extend agriculture into over 1 million acres of previously unproductive land. So that's a really significant achievement in terms of being able to take what's out there and match it to the existing environment or to your own environment. And the figure on the left-hand side is just showing the importance of symbiotic nitrogen fixation in Australia. And you can see, particularly in the southwestern corner of Western Australia, that those levels of the percent of N that's contributed by SNF, or symbiotic nitrogen fixation, can be up to about 95%. So a very large portion of that fixed N is directly coming from these symbioses that we've established. Okay, so one of the big things is, can we predict whether a particular microsymbiont is going to be very effective in the field? Can we predict that in terms of a genetic sense? And certainly genome data will help there. We'll be able to start to look at all sorts of things. Some of the big problems that we have at the moment, including saprophytic competence, or how well strains can actually survive in their environment, and can they persist from one year to the next? That's a very big problem for us. It's one thing to continually sow seed, which has been inoculated with your strain, versus if you can have a very saprophytically competent strain, you can sow it once and then you can just sow your seed year after year without worrying about producing an inoculant. The other big problems that we have include competitiveness. So we see with the introduction of new inoculants that the edaphic ones, the ones which have been naturalised and are used to the edaphic conditions, tend to outcompete the introduced inoculants. This is a very big problem. Quite often those naturalised populations will be more effective at getting into the nodules, but they tend to be less effective in terms of nitrogen fixation. So that's another huge issue for us that we're trying to overcome. Host specificity is another one. What dictates whether one strain can interact with one host versus another with another one? And that can be at the generic level. It can also be at a highly specific level. We're also interested in the infection processes and ultimately in terms of can we predict productivity from some of the information we generate from these genomes. So at the time that we started this work, there were only 5 root nodule bacterial genomes available, and that wasn't much to go on. A lot of those were completely established as models, as microsymbiont models, but we began to soon realise from some of the work, and I'll show you some of that work, that those resources weren't enough for us to predict what was happening in terms of the prediction of whether an isolate can be a superior performer or not. So we needed a more solid resource. And the initial process was to actually do sequencing of 3 isolates. One of those was a Cynorhizobium isolate, Cynorhizobium medici, This is an isolate which can infect and fix with medics. And it's a very acid-tolerant isolate and was used extensively to increase pasture production in southwestern Australia. And the other two genomes were two Rhizobium trifolii genomes. These two genomes were chosen on the basis of biogeographical distance, one coming from the Mediterranean basin, the other from South America, both very distinct host ranges when you're looking at different types of clovers. And certainly with the two Rhizobium trifolia, we begin to actually show now that there's a direct selection by the host. And the plant host seems to be actually selecting out the more effective microsymbiont. So we're very interested in that whole process of selection. So the way that we began to think about this was to actually sequence using a functional genomics vector. And this provides a really powerful resource. You'll see a lot of talks on next generation sequencing, and we've certainly applied that as well to the GEBA project. But in the first instance, this was a very powerful approach because you could set up a couple of model systems, and very rapidly by sequencing out from the vectors, you could create libraries, you could put them into arrays, each vector containing a single insert. And then by actually sequencing all of those inserts, you can backtrack to a particular construct if you want to work with a particular gene, or you can even work in a more randomized fashion because these vectors can be mobilized into the root nodule bacteria. You'll see on the left-hand side here, we've got a coli-1 origin of replication, so effective in E. coli, but not effective in the root nodule bacterium. So these act like suicidal vectors. And because they're suicidal, you can actually now create single crossover insertions. And because your insert is flanked bidirectionally by promoterless reporters, so GFP and LAC-Z in one direction, and GUS-A and T-dimer in the other direction, you can begin to start to look at infection processes and induction and gene regulation. And you can do that in a whole broad range of backgrounds by also creating co-integrants. So you can very rapidly, so once you've done your triparental mating, you can do a single crossover insertion, you can look at expression, you can create a knockout, you can create more than one gene in terms of knockouts because you can use the FRT system here. So the FRT system, by doing a single crossover insertion, you generate one FRT site in the genome. You can then remobilize another plasmid inside. You can generate another FRT site. And all of the intervening DNA you can cleave out using the flipase or the FLP recombinase. So a very powerful way to rapidly generate knockouts, but you can take those plasmids as well, and you can now create the co-integrants by using the ATTP site present in the suicidal vector. You can then mate it with an E. coli strain containing an ATTB site, and together you create a fusion plasmid or your co-integrant, and that fusion plasmid now becomes a broad host range plasmid, and you can now mobilize that into root nodule bacteria, and you can mobilize that into as many different genetic backgrounds as you want. So this was all capable by doing either triparental mating for the single crossover insertions or a single quadruparental mating for the co-integrant formation and mobilization of your broad host range. So a very, very powerful system for us to begin to look at targeting genes and looking at expression. And for the rest of the work that I present, those are the tools that we've actually used. So it was more than just looking at the importance of genomes to the work that we're doing in Australia, but also broaden that right out to the community. And it was a great opportunity to be able to do that. And so what we've done is to set up a resource called the GEBA-RNB resource, or the Genomic Encyclopedia of Bacteria and Archaea for the root nodule bacteria. So it's a sub-branch of the GEBA program. And the idea was to provide a really in-depth coverage of the genomes and to begin to start to apply some of the information we have on edaphic constraints and begin to look at what might be actually constraining species to particular areas. And one of the ones I'm going to hone in on is the effect of acidity and acid pH and determining where cells will be. So the idea, or the approach, was to really take 100 strains and begin to do a deep sequencing approach. These are the strains on the right-hand side here. You can see we've got representatives on the alpha proteobacteria, and we've also got representatives. These are very novel isolates which have recently been discovered in the beta proteobacteria. And The idea being, in the first instance, to send out a call to the international community. I did that using international congress lists and contacting existing collaborators. And we ended up with a total of just over 30 collaborators from 15 different countries. And we put out an invite to submit their favourite organism. We got 300 submissions. And then the process was to whittle that down to 100 of the most important ones of those. And that's just a map on the left-hand side showing the distribution of those isolates across the globe. Some of the criteria we applied to whittle that down to the 100 included that we needed all the associated metadata, so host ranges. We needed to know survival, where they were obtained from, latitudes, longitudes, everything else, so that we could get a good comprehensive picture of these microsymbionts when we started to look at the genome level. We needed good representation of very important isolates, ones which are important for crop and pasture production. We also wanted to cover some very novel isolates as well, some of the new ones that had been recently discovered. A big task was to identify isolates which were not renamed. That sounds easier than it is. Everyone tends to like taking their own organism and relabeling it in their own lab using their own system. And so a real battle was to actually identify the strains so that we didn't end up sequencing things multiple times. And all of these isolates which were suggested to be important had to be NOD plus or nodulation positive, and they had to be fixation positive as well. So from the 100, we then got the collaborators to send the isolates and the DNA through to the Centre of Rhizobium Studies in WA at Murdoch University. And from there, I coordinated sending the DNA to JGI after doing 16S reads, just to make sure that, first of all, that the cultures were pure and that we had the correct genus and species present. For isolates which came from Canada and America, I liaised with Peter van Berkem at the USDA because I felt it was a little bit silly to actually send the isolates all the way from America to Australia and then back again. So he controlled all of those which were from Canada and America. And he did the 16S sequence reads for those and then sent the DNA through to JGI. All of that information is available through the web portal, through the Microbial Genomics program. You can link out, you can find out the current status. We've done 89 of the organisms so far. So we're about 89% through the actual project. And you can see all of the status of whether the organisms are finished or whether they are still in the production line and so on. You can link out on the informatics and go through to IMG. You can go straight to IMG if you wish. And all of the data that we have so far is present there as well. Okay, so what I want to do now is come back to those edaphic constraints that we were talking about and start to apply some of the information we've learned from the genomes. And here I think this puts us into real perspective. These are our sandy, very acidic soils that we have in WA. And so what you do here is what we call a crossroad technique when we're selecting out strains for superior characteristics. So this would be a test for a strain which could survive in those acid conditions. So in the middle of the plate, you put your inoculated seed in year 1. And then you let that grow out for 1 season, and then year 2 you come along and you sow uninoculated seed across. And what you're looking for is a generation of pasture. Okay? So you can see there that that strain is very acid sensitive and it's no good in that environment. Here's a strain which can persist at the original site of inoculation, okay, but it's not saprophytically competent and it can't handle those acidic conditions. And then here we have an isolate which is very effective and can spread right across the crossroad, very saprophytically competent and very acid tolerant. And that particular isolate there is Cynorhizobium medicae WSM419, which can nodulate and fix well with metacargo murex. So that's actually metacargo murex on that plate photo there. So a lot of the work is in a practical environment, doing a lot of the work in field trials and in glasshouse tests. And that's all very well and convenient, but it'd be much nicer if we could try to get a handle on some of the genetic potential of these strains and to be able to identify that from the genetic pool so that we can begin to enhance our success rate in developing superior inoculants. So one of the ways we've gone about this, certainly in the beginning, was to look at all of the genes inside the cells which are important for survival in an acid environment. We also looked for genes which are expressed by acidity. Here's one of those. This is the most acid-induced gene that we've discovered so far. This one we call the low pH-induced gene A, or LIPA. And here you can see it's a link to a gusso fusion, so exactly the process I talked to you about earlier using those vectors. You can see the amount of induction here, 20-fold at pH 5.7, so very massively induced. I'll come back to the other two backgrounds in just a moment up the top. But you can take that particular construct, you can sequence out from that plasmid, you can define the genes that are present there, you can take that to the genome, you can clip out that whole region from the genome, and you can begin to actually work out the roles of each of those genes, which we've done. You can actually take those resources that I talked to you about with that vector system, and you can knock out every single gene, you can look at the expression of every single gene, and we've done that for this region as well. So you'll see here, downstream from LIPA, we've got ACVB, which is an acid-induced gene, which is also required for virulence. And then upstream, we've got 3 genes which are part of 2-component signal transduction systems. So we've got a sensor here, we've got a regulator just downstream from that, and another fuse sensor regulator downstream from that. And you can create knockouts in all of those. You can investigate the expression of LIPA in each one of those. And then we can also look for any other factors which can control expression. And certainly you can find the sigma factor, which is required for expression. So you can see the decrease in this RPN negative background compared to the wild type. And associated with RPN, you have enhancer binding proteins. And by fishing out all the enhancer binding proteins, knocking all of those out, you can begin to identify the enhancer binding protein which works with this particular sigma factor to control the expression of LIPA. So we've done a lot of background work to look at genes which are essential and critical for this survival in acidic conditions. And here's our regulatory model so far. So we've actually built up a quite comprehensive model for, first of all, recognition of the acid signal through the histidine sensor kinase, transfer of that signal through by phosphorylation to 2 regulators. One of those is the TCRA protein. The other is the fused sensor regulator protein, or FSRR. And by phosphorylation steps, eventually you get through to the phosphorylation of the enhancer binding protein, and that's for the dimer form. And with phosphorylation, you get the hexameric form. And with that hexameric form, we can now show that we can get binding of RNA polymerase to the very specific promoter site. We've even identified the transcriptional start site for these genes. And we can show the interaction that's occurring between these particular proteins. So now we can begin to explore a little bit more about what the role of this protein is. So if you have a quick look at the databases and you just do a blast search, what you find immediately is a number of unknown domains, so that's not particularly useful in itself. But embedded in this is a lyase domain, which is potentially involved in the transfer of lysine. If you take this protein and you blast it across into the databases, you come up with a huge number of hits. And what you find is that this particular protein aligns with proteins which are found predominantly in pathogens and in symbionts. And what I've done on this particular map is just to highlight in certain regions where people have actually performed experimental evidence and shown which particular amino acid is being transferred. So by all accounts, it would appear that this particular protein, the LIPA protein, is a potential amino acid phosphatidylglycerol synthetase based on the homology with some of these proteins. And we're postulating at this point that this particular protein can transfer lysine. And so you would predict, based on homology with the other systems, that a potential RNA synthetase is involved. So now you can ask the question with the genome resources that we have, what is the distribution of this particular gene? Are there regulatory networks in place in all of these other genomes? And what is that telling us about the system? So if you just quickly look on the left-hand column, you'll see all of the genera that we've sequenced, and in brackets the number of genomes that are represented. And I've thrown in Staphylococcus down the bottom because in Staphylococcus we have the MPRF homologue for LIPA. And a lot more is known for that in Staphylococcus than in any other organism. But certainly what you can first see is that LIPA is not distributed in all of the root nodule bacteria. And the second thing that you can note in the second column is that one of the main regulatory elements, FSRR, is actually present when LIPA is actually present. Okay. Now, it was of interest, based on the homology to the lysine phosphatidylglycerol synthetases, to postulate that potentially there is a lysine tRNA synthetase involved in the system. So what I've done is I've looked at lysine tRNA synthetases and their distribution in these genomes, and you can find something very interesting. We've got two predominant forms in the root nodule bacteria, and this was a bit surprising. The type 2 form is the most common form, and it was discovered after the type 1 form, and the type 1 form is actually a very rare form. It's found to occur in less than 10% of bacteria that have been sequenced so far. And yet, if you have a look in the root nodule bacteria, there's a very good representation of this particular tRNA lysyl synthetase. What we've done then is to actually look at matching any other cogs that are present from the genome data, and we actually found one cog which aligned perfectly with the presence of the rare form of this lysyl tRNA synthetase. This cog is 1494, and you can see an absolute correlation in all of these genomes that we've sequenced for this cog, for the presence of this cog when you have the presence of that rare form of lysyl tRNA synthetase. So this is very interesting at this point because it's the class 2 form which can actually take the free form of lysine and it can incorporate it utilizing ATP. So the important thing here is that these lysyl tRNA synthetase types are similar, but actually they've got very different roles. And what you can see here, this is work done by Herve Roy at the University of Florida. He's actually looked at analogues of lysine, and he's asked the question, is there any effect when you look at the lys RS1 type or the lys RS2 type? And the answer to that is yes. So with the lys RS2 type, you get sensitivity to an analogue like thiolysine, and the lys RS1 type, you don't get that sensitivity. And if you look at something like GABA, which is down the bottom here, with GABA, you get the reverse situation. Okay, lys RS1 is sensitive to GABA, but it prevents inhibition by thiolysine. And this is very interesting because there's been a report out which suggests that certainly for metacargo truncatula, GABA is very important in upregulating the amount of nitrogen that's fixed. Which comes finally to the role of GABA in the whole system in regulating nitrogen fixation. It's now been postulated that GABA is a very important compound which is being used by the plant to sense the amount of nitrogen which is being fixed. And so this is just a postulated model of the actual metabolism that's occurring inside the nodule. This is a bacteroid over here inside that nodule. And by increasing the amount of GABA, we're increasing the amount of metabolism feeding into the TCA cycle, and we're increasing the amount of compounds which are being secreted out, nitrogenous compounds, and these are being back converted into GABA. So the amount of GABA that's present in the nodule is critical to tell the plant how much nitrogen is being fixed. If you actually correlate that with different plants, this is just data that's currently known. You can see in Metacarga sativa, you've got very high levels of GABA, as with Trifolium repens. But if you look at Glycine max and Lupinus, you can see that those levels are very low. And interestingly enough, that correlates with the presence of lys RS2. So now we can postulate that lys RS2 might in fact be critical in establishing the chromosomal type which is present in a nodule. Okay, so just summarise then. This resource will become incredibly important to the community to begin to look at all sorts of different edaphic parameters, survival and persistence, competition, what are the reasons governing why one strain can fix more effectively than another. More importantly, we can now start to look at identifying markers to identify superior inoculants. And this particular study has now highlighted that for the root nodule bacteria, actually strains can have lys RS1 or lys RS2, okay, either one or the other. And in fact, you have a very big predominance of the rare form of lys RS1 in the root nodule bacteria. Lys RS2 might be important for incorporation of free lysine into tRNA, and that might drive the enzymatic activity for this lysyl amino acid phosphatidylglycerol synthetase. And this work has highlighted that potentially this now provides an important suggestion as to why nodule environments might be dominated by certain types of root nodule bacteria, in that that lyase RS type would dictate the presence of a particular organism based on the amount of GABA that's present in that nodule. And just to finalise, I'd like to thank all of the people involved in that work, and I'd like to leave it at that point. Thank you.
Okay, well, there are a lot of organisms in the same genera as these root nodule bacteria that are both plant-associated and non-nodulating, and others that are free-living. Do you think that generating a lot of those genomes as well is going to help you interpret what you're learning from these root nodule bacteria? Or?
I think it very much depends on the question that we're asking. So if we're looking at maybe survival in a rhizosphere environment, definitely that could really help. If you're looking at what's driving the basis of metabolism inside a nodule environment, then I think that perhaps would be less important in that regard. But certainly by understanding more clearly the roles of organisms in the rhizosphere, that's very, very important. Mm-hmm.